welcome back to your classes on overview and integration of cellular metabolism. We are in week 11 and class 52, and this is the third class of nucleotide metabolism, where we will be covering pyrimidine metabolism and its associated disorders. Specifically, we will be talking about pyrimidine chemistry, we will be talking about the de novo synthesis of pyrimidine, we will be discussing about thymidylate synthesis, how nucleotides are reduced into deoxy form. We will be talking about the regulation of pyrimidine biosynthesis, the pyrimidine salvage pathway, and lastly we will be discussing about disorders of pyrimidine metabolism. All right. So, to start with, these are the major pyrimidines. We discussed about purines in our previous classes. These are the pyrimidines, and regarding pyrimidines, the chemistry is very easy. There is only one ring. In purine, there was a hexagonal ring followed by a pentagonal ring. Okay, I hope you remember that. In case of pyrimidine, there is only one benzene like ring, so with 6 sides, and the numbering we will start from the first in the nitrogenous carbon and go in a clockwise direction from 2 to 6. All right, and different pyrimidines, that is cytosine, uracil and thymine, vary in their side chains. For example, here between cytosine and uracil, this ox or keto group has been changed to an amine group, right? And between uracil to thymine, you can see there has been addition of one methyl group. So, these methylation, amination, these are common things that you should keep in mind when we are dealing with interconversion of pyrimidines, right? So, this concept will be applying later in our lectures. Now, this is the same slide that we shared in our purine metabolism classes. We showed how these are the nitrogenous bases. For this class specifically, we should focus on these three, all right, uracil, cytosine and thymine, because these are pyrimidines and these are their respective nucleoside form. Nucleoside means nitrogenous bases and a ribose sugar, and these are the nucleotides. Nucleotides mean ribose sugar, nitrogenous base and a phosphate group. So, all of them have got a phosphate. So, it can be monophosphate, that stands for AMP, or diphosphate DP, or triphosphate TPs. For example, UMP, UDP, UTP, those all are nucleotides, all right. And the deoxy are the bases that are, deoxy are the variety of sugar that belong to DNA, and ribo in RNA, that you already know, all right. So, like purine synthesis, if you understood that class very well, it is very easy to follow in this class because it involves the same concept just like purine synthesis. So, there are two ways of synthesis. Number one, de novo. De novo means starting new. So, we assemble a car by selecting absolutely factory made new parts. All the new things one by one gets assembled to make a new car. This is the analogy. And in case of salvage pathway, it means we are using already used up product that are almost pre-made, and we just assemble that car using pre-made parts from used up or other cars, all right. So, this is the analogy of salvage paths. So, we are using basic nucleotides to synthesize, or basic already synthesized pre-nucleotide to synthesize their monophosphate or triphosphate versions, all right. But in case of de novo synthesis, the ring will be synthesized, each element will be added one by one, but in salvage pathway the ring is already there, the nitrogenous base is there, we simply add the phosphate group to functionally achieve the nucleotide synthesis, right. So, in case of pyrimidine biosynthesis, what are the components, who are the donors to form the ring, right? Very important MCQ question. Glutamine, carbon dioxide in the form of bicarbonate, and aspartic acid or aspartate. Okay, so, these are the three components who actually donate the atoms that form a pyrimidine ring. There is a difference between pyrimidine synthesis and purine synthesis. You saw in case of purine synthesis, the first step was when PRPP was formed, right? PRPP, that is phosphoribosyl pyrophosphate. So, basically PRPP provided the ribose 5-phosphate group on which the entire purine nucleotide was formed. Here it is completely different. This approach in case of pyrimidine synthesis, here first the ring is formed and only at the very last step, at the later stages, ribose 5-phosphate is added to it. Okay, so, this is the difference. In case of purine, first ribose is attached and on ribose the entire ring is formed. In case of pyrimidine, first ring is formed and then ribose is attached, all right. So, in case of prokaryotes, the total synthesis of pyrimidine happens in 6 step. This is a 6-step pathway. Why I am saying prokaryote? Because in eukaryote the system is much more simplified, right. So, let us discuss the essential first, because most of the questions are framed keeping prokaryotic system in mind, especially in case of pyrimidine biosynthesis, all right. So, we will break down all the steps one by one. So, let us look in the first step what happens. Glutamine and aspartate, I am sorry, glutamine and carbon dioxide in form of bicarbonate forms carbamoyl phosphate, okay. So, you have heard this name carbamoyl phosphate, where? I hope you remember, right, because there I said carbamoyl phosphate we will again encounter during pyrimidine biosynthesis, because we discussed the enzyme carbamoyl phosphate synthetase, right, that was 1. Here we are discussing about carbamoyl phosphate synthetase 2. So, this is the first step that is catalyzed by the reaction carbamoyl phosphate synthetase 2, where glutamine and carbon dioxide forms carbamoyl phosphate, right. And you will see that ultimately its carbamoyl phosphate extends to form a ring. So, basically we can say that immediate precursor of this nitrogen and carbon is not glutamine and carbon dioxide or bicarbonate, it is actually carbamoyl phosphate, all right. So, please remember it is actually carbamoyl phosphate and aspartate that is donating all these. In reality, the carbamoyl phosphate is again formed from glutamine and carbon dioxide or bicarbonate. So, you get the answer. So, if you get a question, all of these are sources of atoms in case in pyrimidine nucleotides, and you get an all except type of question. Remember, there are 4 correct possible answers: glutamine, carbon dioxide, bicarbonate, aspartate and carbamoyl phosphate. If carbamoyl phosphate is an option, you cannot mark it wrong, because carbamoyl phosphate is also actually a donor of the atom in case of pyrimidine ring. Very important for exam point of view, right. Next step, what happens? This carbamoyl phosphate combines with aspartate, another donor of the pyrimidine nucleotide ring, and it forms carbamoyl aspartate. The enzyme is aspartate transcarbamylase, which is abbreviated as ATCase or ATCase, aspartate transcarbamylase, all right. So, carbamoyl phosphate and aspartate forms carbamoyl aspartate. You can see the ring is almost formed. So, what else is left? Simply we can join the ring, and that is what exactly happens. One water molecule goes out, you can easily see, we can easily let go of one water molecule from here, right. So, one water molecule will leave and lead to the formation of dihydroorotic acid, which remains as a salt in medium by combining with cation, and also known as dihydroorotate. So, dihydroorotate, dihydroorotic acid, it's same, all right. Many textbooks will have different names, and the enzyme is dihydroorotase, right. This is the ring closure step. What happens in the next step? Dihydroorotate is converted to orotate, or dihydroorotic acid is converted to orotic acid, all right. And the enzyme is dihydroorotate dehydrogenase. What Q and QH2 stand for? This is a reducing equivalent that is actually helping in the dehydrogenase reaction, okay. So, simply there is a double bond formation over here, okay. This is the NADH dependent dehydrogenase, okay, and it leads to the removal of hydrogen atoms from the single carbon atom, right. So, we get orotate or orotic acid. In the next step, what will happen? So, once we got the ring, as I discussed, now it will be added to the ribose 5-phosphate, and we already read who is the donor of ribose 5-phosphate in all purine and pyrimidine de novo pathway. Yes, you are right, you have guessed it right. If you have guessed PRPP. So, PRPP, so the enzyme is orotate phosphoribosyl transferase, and it forms OMP, orotidine 5' monophosphate. So, basically what happened? Entire ribose phosphate molecule has been attached to the ring, okay, and PRPP gets converted to pyrophosphate PPI, which again hydrolyzes to form two molecules of inorganic phosphate. All right. Remember, you should be able to connect this to the very early class of thermodynamics, because all of these reducing equivalent hydrolysis ultimately makes these reactions endergonic. All right. So that they can spontaneously happen, okay, exothermic. Next, so what happened? We got OMP, orotidine monophosphate, means we got a ring, we got a phosphate group, and so what else is left? The main thing is OMP is not a familiar pyrimidine. So what are the familiar pyrimidines? Cytidine, uridine and thymidine. So our main goal is this pathway can only stop if we achieve any one of those three. So this is the main final end product in a 6-step pathway, where OMP, by the action of a decarboxylase enzyme, simply this carbon dioxide will be replaced by a oxo group or keto group. It forms UMP, uridine monophosphate. Okay. So UMP is the final product of de novo synthesis of pyrimidine in prokaryotic system. So why I am saying repeatedly in prokaryotic system? Mind it, the system is actually same, the mechanism is actually same in case of eukaryotes, even in humans, right? But in eukaryotes, though there are multi-enzyme, the proteins have got multi-enzyme domains. For example, step 1 to 3 is catalyzed by a single protein, that is known as dihydroorotate synthetase. So one substrate goes on and ultimately we actually get dihydroorotate synthetase, okay. And step 5 and 6 are catalyzed by a bifunctional enzyme, UMP synthetase. So there is no separate phosphoribosylate transferase and decarboxylase. So step 5 or 6 are again clubbed together in case of eukaryotes. So there are mainly 2 enzymes in eukaryote which play the role of 6 steps, okay. 4th enzyme is there, okay. So in eukaryote there are total 3 enzymes. Next, so we got UMP, right. So our known pyrimidine, UMP, uridine, cytidine and thymidine. So this is a slide that shows how we get cytidine from uridine. So first what happens, this is done in 3 steps. So UMP is acted upon by kinase. It is first converted to UDP by UMP kinase, and then by UDP kinase it is converted to UTP. Thereafter it reacts with glutamine to form CTP, okay. So it can be considered a step 7, 8, 9, okay. Anyway, so if we just highlight this step, okay, these are 7, 8, 9 steps, and if we highlight this last step, you can see that what exactly is happening here. This keto group is being converted to an amino group. This is the basic difference in structure of uridine and cytidine. And who actually helps in donation of this nitrogen? Glutamine. All right. Again, ATP is required, hence this is known as synthetase enzyme. So CTP synthetase is the last step. So 2 kinase followed by a CTP synthetase, right. So next what? So we got UDP and CDP, right. So next step, before going any further, we should know, we did not discuss in purine synthesis because the concept is same. So you should know that till now we actually discussed synthesis of ribonucleotides, but we, for DNA synthesis, we need deoxyribonucleotides. So who does this? By simply reduction. A reduction reaction, it is done by the enzyme ribonucleotide diphosphate reductase. It is also simply referred as ribonucleotide reductase. Mind it, nucleotides are reduced in diphosphate form only by this enzyme. So ADP is converted to deoxy ADP, CDP to deoxy CDP, GDP to deoxy GDP, and UDP to DUDP. All right. This concept should be very clear. Diphosphates are the substrate for ribonucleotide reductase. That is why this enzyme is also referred to as ribonucleotide diphosphate reductase or RDR. All right, diphosphate, very important. But this is not simply, I mean, this is not as easy as it seems. Just a reduction, yes, it is just a reduction, but there are many things that are going on behind the hood. So what is that? You see, ribonucleotide diphosphate reductase, actually what it does, it simply removes this oxygen from the 2' position and it converts this ribonucleotide diphosphate to deoxyribonucleotide diphosphate. Water molecule is removed. Okay, it is done by the enzyme, as I told you, ribonucleotide reductase. Well, ribonucleotide reductase has been shown, has been studied to be active in reduced form. I mean, there are disulfhydryl groups in the enzyme that catalyzes the reaction that are active when they are in reduced form. So, and during the reaction, that is ribonucleotide reductase reaction, they are converted to their oxidized form. Okay, you can visualize this analogy with glutathione, right? There was also 2 SH bond, we are reducing it by glutathione reductase. So much more, I mean much like that. So ribonucleotide reductase is now inactive, it is in oxidized form. So we need to again reduce that, so that action of, I mean activity of ribonucleotide reductase is restored. Who does that? It is done by another protein that is known as thioredoxin. So thioredoxin helps in reduction of ribonucleotide reductase, all right, to convert it from oxidized form to reduced form. So by doing that, thioredoxin also gets oxidized, fine, but we need reduced thioredoxin to catalyze this reaction. So who will reduce thioredoxin? The enzyme thioredoxin reductase, all right. So again, thioredoxin reductase is also active in reduced form, okay, and when it does its action, it is inactivated and it is converted to oxidized form, and finally it is also regenerated by getting converted to reduced form with the help of reducing equivalent FADH2. FADH2 gets converted to FADH, ultimately who helps in regeneration of reduced FAD to FADH2? NADPH. So, there is a big chain of redox coupling going on here. So, you can just pause this video right at this moment and you can visualize, think calmly, you can easily understand. Reduction of this substrate, all right, this gets reduced. This enzyme, the first enzyme ribonucleotide reductase, helps in its reduction, itself gets inactivated and oxidated. It needs to be further reduced. Who reduces it? Thioredoxin. Someone which will reduce any other substance will itself get oxidized, basic redox principle. So we got oxidized thioredoxin. Oxidized thioredoxin again needs to be reduced, it is done by the enzyme thioredoxin reductase. Thioredoxin reductase itself gets oxidized, we again need to reduce it with the help of FADH2. FADH2 will also get reduced, so NADPH helps in reduction of FAD to FADH2, alright. So if ribonucleotide reductase comes as a short note in any descriptive exam, be prepared to draw this diagram and you should practice it on your own so that you can reproduce it in the exams, okay. So now we will discuss how we can generate or produce DUMP in our system. We discussed ribonucleotide reductase. So whenever we are discussing about deoxy sugars, ribonucleotide reductase is producing deoxy sugars, but it is dinucleotide, but we need mononucleotide. Why? We will see, but right now we are focusing in production of mononucleotide, that is deoxymononucleotide, which can be done in two ways. Number one, an ADP molecule can combine with DUDP, it forms DUMP plus ATP, all right. The second way, where DUDP combines with ATP to forms DUTP and ADP, right. and in the second step DUTP gets hydrolyzed to form DUMP and pyrophosphate. So these are the two ways how we can generate DUMP in our system. Why are we so much focused regarding DUMP? It is because we need DUMP to form thymidylate, right? The last pyrimidine, the last piece of puzzle that was unsolved. So we got UDP, we got CDP, now we get thymidine. Thymidine was not formed, right? Thymidine, to form thymidine we need DUMP, because this is the main component which is needed for DNA synthesis. Okay. Now this DUMP is acted upon by the enzyme thymidylate synthase. Okay. We will break it down. So this is a reaction in a nutshell. Okay. Why this reaction is important? Because once we have studied the whole reaction, we can answer the question that the situation that we studied when we were discussing nucleotide analogues. We studied nucleotide analogues like 6-mercaptopurine, 5-fluorouracil methotrexate, and I told you methotrexate was inhibiting the enzyme dihydrofolate reductase, right? 5-FU or 5-fluorouracil was inhibiting thymidylate synthase. So these are the enzymes that are the target areas for chemotherapeutic drugs or anti-cancer drug, right? So just remember dihydrofolate reductase and thymidylate synthase. So now let us break this reaction. Let us magnify the steps. So we got DUMP from two ways in last slide. So DUMP, how can we convert DUMP urid into thymine? I showed you during the in the very first slide of pyrimidine by simply adding a methyl group. So this is nothing but a methylation reaction done by the enzyme thymidylate synthase, DUMP to DTMP. Who is the methyl donor? Common methyl donor, we came across this molecule in multiple area, N5N10 methylene tetrahydrofolate. It is often also abbreviated as 510 or often it is written as N5N10. N5N10 methylene tetrahydrofolate itself it is converted to dihydrofolate. You can track the structure if you are interested to see what molecules are getting transferred or from where exactly the methyl group is getting transferred. It is this one and this hydrogen, they are getting transferred to form DTMP. Our job is done. So what do we need more? We need more N5N10 methylene tetrahydrofolate so that multiple DUMP can be converted to multiple DTMP, right? So we need regeneration of N5N10 methylene tetrahydrofolate. So who does that? So we got dihydrofolate in the previous reaction. This is acted upon by the enzyme dihydrofolate reductase to form tetrahydrofolate. The reducing equivalent who donates the hydrogen group in order to convert dihydrofolate to tetrahydrofolate is again NADPH. Okay, and I told you this dihydrofolate reductase inhibitor, for example methotrexate, is an anti-chemotherapeutic drug, right, or chemotherapeutic drug or anti-cancer drug. All right. So we got tetrahydrofolate. Now this reaction, tetrahydrofolate to methylene tetrahydrofolate, again there is a transfer of methyl group. Basically this accepts a methyl group to regenerate N5N10. Who donates the methyl group? If you can connect to your previous class where we discussed synthesis of serine and glycine, SHMT, serine hydroxymethyl transferase. Over there we emphasized on this part and we just mentioned these molecules, right, and I told you when you finally know this reaction of pyrimidine synthesis, the entire 1-carbon reaction chain will be completed. So I hope you can now open that class or go back to that class after you are done with this class, and now you can fully relate this entire 1-carbon reaction cycle. Well, let me tell you, this is an opportunity or this is a must. This is a time where you should go back to all of your classes, because very soon the integration of metabolism classes are coming up, and for that you need to have all of the metabolic cycles at the back of your mind so that you can easily connect and relate to that class without pausing that video 1000 times, right. Moreover, the upcoming class will be about inborn error of metabolism, which will also require you to revise various disorders of metabolism that was discussed in throughout the various weeks. All right. So, this is a very good time to start revising if you have not started it already. Okay. So, let us move on. So, let us discuss about carbamyl phosphate synthetase. I told you, where did you remember? Did you remember carbamyl phosphate synthetase 1? If you remembered urea cycle, I give you 10 upon 10 because urea cycle is the one where carbamyl phosphate synthetase 1 was discussed. Over there, CPS 1 was utilizing an ammonia, a bicarbonate and 2 ATP molecule to form carbamyl phosphate, whereas in case of CPS 2, as we studied, it is using carbon dioxide or bicarbonate, glutamine and forming carbamyl phosphate by using ATP, right. ATP is used because these are synthetase enzyme. This is variety 2. Now, how these are regulated? Okay, CPS2 is allosterically regulated, right. So, who are the allosteric activators? PRPP and IMP are allosteric activators, and several pyrimidines, UTP and CTP are allosteric inhibitors, right. You should know that this enzyme aspartate transcarbamylase is often, although this is the second step of the pathway, this is actually the first committed step of the pathway, all right, and multiple regulators also affect this step. So, positive regulators are ATP and negative regulators are UTP and CTP. So, can you relate how UTP, basically mainly UTP, right. So, UTP is acting as a feedback inhibitor and by default CTP because we get CTP from UTP. How it is acting? Simply you just visualize, if this reaction happens, we got multiple UTP in our system. So, if our body has got more and more UTP, we do not need to synthesize any more UTP. This is simply a step of or concept of feedback inhibition. Okay. So, here is a actually more schematic diagram. If this regulation of pyrimidine nucleotide comes as a long question, you can practice this diagram and represent, so that we can see CTP and UTP are inhibiting aspartate transcarbamylase. ATP is actually helping or promoting the action of aspartate transcarbamylase. Also, you should keep in mind CTP inhibits its own synthesis by feedback inhibition, and GTP actually helps its own synthesis allosterically. Okay. So, the basic difference between purine and pyrimidine nucleotides, as I told you, they are synthesized on PRPP, ribose molecule, on top of it is added, whereas it is first synthesized and then added to PRPP. It is regulated by reciprocal regulation. I hope you remember GTP and ATP, it is mainly regulated by UTP, it generates IMP, here it generates UMP and CMP, and both requires energy, right. This is not a difference, we are just comparing and contrasting. Also, one important information regarding ribonucleotide, reductase, that is formation of deoxyribonucleotides. This is a chart. If you actually want to score very good in your exam, this is something you need to remember, because it is difficult. Number one, it is difficult, and this is a very nice to know area, specifically because you need to remember so many permutation and combination of molecules. So, who is actually acting as a positive regulator and inhibitor? There is no easy way of remembering this. You just have to write this over and over again to commit it to your memory, right. So, we will now discuss the breakdown of nucleic acids. Concept same, just like purine we discussed. So, nucleic acids will be first broken down by nucleases to mononucleotides, right, and then it will be broken down by nucleotidases and phosphatases to nucleoside, which will be further broken down to nitrogenous bases. Now, this nitrogenous bases can be degraded by direct catabolic pathway, or they can be reutilized using salvage reaction to form 5' mononucleotides, right. So, in salvage of pyrimidine, we can see basically here a case where uridine has been converted to uracil, that is the base, and ribose 1-phosphate, and in this salvage reaction uridine is directly combining with uracil is combining with ribose 1-phosphate to form uridine. This is a reversible reaction, alright. It can occur in both ways. Here we can see interconversion of multiple pyrimidine by cytidine uridine kinase, here also deoxycytidine kinase, thymidine kinase. So, all of them convert these bases to their monophosphate forms. And I mean, this is very obvious, alright. Interconversion of pyrimidine nucleotides, you can see first we are forming a uridine product, then by reducing it we can form deoxy, DUMP actually, and by methylation we are forming thymidine, right. Again by transfer of amino group from glutamine we are getting cytidine, and then we can again reduce it to form deoxycytidine, and similarly they can be converted to their mono and di and simply by kinases and phosphorylase enzymes into their phosphorylated less and higher phosphorylated forms. Okay. And just as a revision, I hope now you can easily understand the conversion of multiple purine nucleotides in a similar way. You can just place the central synthesis of IMP by de novo synthesis, and just you can relate how those are interconverted into each other. Okay. So, finally, we come to the last phase of the discussion, that is pyrimidine catabolism. So, pyrimidine nucleotides are hydroxylated, just as we discussed. These are no exception to purines by to their nucleosides and inorganic phosphates. So, nucleoside means ribose and nitrogenous bases, okay, and phosphate, once we detach it, nucleotide becomes nucleoside. On further degradation, thymine, uracil, those individual nitrogenous bases will be produced. So, our goal is to determine how this thymine and uracil bases are degraded, and upon degradation they will produce intermediates of central metabolism. What is central metabolism? Basically the integration of metabolism, and we will be able to identify certain common products by which you can easily say where those common products can be diverted. Okay. So, this is actually the pathway. So, again let us break it down, magnify each and every pathway. So, we start with uracil and thymidine. They are first acted upon by a dehydrogenase enzyme, NADPH dependent dehydrogenase enzyme, that will be converted into dihydro version. So, dihydrouracil and dihydrothymine, which will be acted upon by the enzyme dihydropyrimidinase. Simply a water molecule is coming and it is converting them to uridopropionate and uridoisobutyrate. Okay. The structures are not important, the names are important. Of course, the structure is important for you to understand conceptually, but it is not important for you to remember and draw all these structures, because it is difficult to remember so much. Okay. But if you can, it is best to remember, because otherwise you won't need to simply mindlessly memorize each and every metabolic step. And lastly, what happens? This uridopropionate and uridoisobutyrate are acted upon by the enzyme uridopropionase, and they form beta alanine and beta amino isobutyrate. These are successively degraded to acetyl coenzyme A, substrate for many things, and ultimately succinyl CoA. So, both of them can enter into the carbohydrate metabolism, or there are multiple fates of acetyl CoA and succinyl CoA that have already been discussed. So, finally, we are discussing about disorders of pyrimidine metabolism, and there is only one that you need to remember due to the deficiency of. So, why there are three alternatives? Because in prokaryotes these are two different enzymes, and in eukaryotes this is basically a single enzyme that forms performs these two steps. So, what happens? Orotic aciduria means orotic acid is accumulated. In the last two steps, orotic acid is converted to OMP and finally to UMP. This is not done. So, this is autosomal recessive disorder. What are the characteristic feature? Definitely orotic aciduria means excess orotic acid will be excreted in urine. Even crystals of orotic acid have been found to be excreted in urine, and those may cause urinary tract infection. Apart from that, what are the systemic symptoms? Growth retardation and anemia, megaloblastic variety of anemia. What is the treatment? Treatment is actually administration of UMP, alright, or uridine triacetate, which is converted to UMP in vivo. So, how actually UMP administration treats orotic aciduria? Simple. If we have got excess UMP, ultimately it forms UTP in system. If there is excess UTP, I told you UTP inhibits the pyrimidine biosynthesis, and if UTP, in fact it inhibits synthesis of carbonyl phosphate. So, if the committed step or the earlier step is actually inhibited, there will be no production of intermediate. So, there will be less and less production of orotic acid or orotate, and hence this will be treated. I mean, there will be no substrate to excrete, fine. So, UMP will act as a treatment for orotic aciduria. And what are the varieties of orotic aciduria? They can be found, I mean, they have been elaborated as multiple types. One is a major type in which both enzymes activities are deficient, that is known as type 1, is much more serious, whereas there is a milder variety in which only the decarboxylase function is deficient. So, there will be OMP, okay. And in the first majority variety there will be formation of excess orotic acid. Mind it, we also mentioned orotic aciduria in urea cycle due to the enzyme deficiency ornithine transcarbamylase. If this enzyme is deficient, what will happen? That will lead to a situation of hyperammonemia along with orotic aciduria, because urea cycle disorder leads to hyperammonemia, but there will be no inborn orotic aciduria. Mind it, these are all inborners, the first two that presents very early. And a very minor non-symptomatic disorder is beta amino isobutyric aciduria. There is a transaminase deficiency, and basically this is non-symptomatic and do not need much treatment to start with, okay. So, this is the conclusion. We have discussed pyrimidine chemistry, we have discussed de novo synthesis of pyrimidine, we have discussed how it is synthesized in prokaryotes and eukaryotes, we have discussed how it is regulated, we have discussed the reduction, how it is reduced by ribonucleotide reductase, we have discussed synthesis of thymidylate, the salvage pathway, and interconversion of pyrimidine, fine pyrimidine catabolism, and the disorder of pyrimidine biosynthesis, that is orotic aciduria. Well, these are my references for the today's class, and I thank you all for your patient hearing.